Guys, I hope you can hear me very well. And today, this quarter, I will be your lecturer. And someone please let me know whether you hear me very well or not, okay? And is it audible, my lecture, or not? And thank you for coming. Today I can see at least some students here already. And this quarter I will be your lecturer. And I would like to tell you that we already finished lectures for general microbiology. And we start now the lectures for special microbiology. And our lectures will be started with the pathogenic fungi. We have touched them a little bit in general microbiology, but in just regarding the way of how do we culture them a little bit and how we identify them. But then today's lecture will be describing really the pathogenic fungi. And usually in our special microbiology lectures, I will follow a particular plan. So the description of the pathogenic microbes, a bit of the structural differences with other microbes. Then how do we categorize them? What kind of diseases they cause? And this is a very important thing. How do we culture them, identify them, and what kind of drugs do we use to inhibit their growth or to kill them? So, and we start on the first slide. You can see a picture gotten in our department in some studies from the human blood. And you can see here the macroconidia of the pathogenic fungi with many spores that germinate and produce sieved or hyphae. You can see here that's because some fungi, pathogenic fungi, when they got inside the human blood, they may start growing very quickly, abnormally quickly, right? Okay, so that's why they form not the regular cells but elongated, very long thread-like structures. Okay, we go ahead with the characteristic of the structural features of fungi. You can see here air cell, and the fungi can be unicellular or multicellular, but it doesn't matter because both of them are eukaryotic organisms. And it means that in many features they look like and function like other eukaryotic cells. And from the very beginning, I would like to underline that how do you think? Is it bad for us or good for us if we've got some pathogens inside our body? And of course, the answer it is bad for us. Why bad? Because the basic structure of the fungal cell is very much same with that of the human cell. And this is not like in bacteria, where the basic structure of the bacterial cell has many differences with the human cell. And when we try to find the drug, we may use these differences in the structure of the pathogen or cells of the pathogen and cells of the host. So in bacteria, we have so many differences. So that's why we have a lot of different types of antibiotics with very different targets for antibacterial treatment. But then if we are talking about eukaryotic pathogenic fungi, they don't have that many differences with the human cells. They have the general plan and structure of the eukaryotic cell. So that's why it is so difficult to find the target for antifungal drugs called also antimycotics. So that's why, generally saying, the anti-fungal or drugs or antimycotics all much more toxic for human body than that of the against bacteria, than the drugs against bacteria, because it is difficult to find the point of selectivity for these antimycotic drugs. I hope you understand my idea. I go ahead with the structure. So every fungal cell or each fungal cell has a cell wall. Can you see? This is a cell wall. Then beneath the cell wall we have a plasma membrane. Inside the cell, we have a cytoplasm with a nucleus where we have chromosomes and with a nuclear membrane, typical for eukaryotic cells. Then, of course, we have endoplasmic reticulum, then mitochondria are there. I just want to remind you that bacterial cells don't have these very well developed organelles, right? So, and then of course, a lot of ribosomes. That is the general picture of the structure of the fungal cell. So, a few words about the structures. So, I would like to start with cell membrane, and you will understand why. So the cell membrane is composed of glycoproteins, phospholipids, and the lipid that is called ergosterol. Then it means that fungal cell membrane phospholipids are different from human cell phospholipids. So, and I would like to underline that fungi have ergosterol instead of cholesterol that is found mainly in the human cell membranes, human cells, cell membranes. Okay? So, and I would like to underline that ergosterol of the fungal cells is used widely as a best target for selective antimycotic treatment, as a target for antimycotic drugs. So this is about cell membrane. Now we go to cell wall. It is a very complex structure and it is a very rigid biopolymer, and of course it is responsible for the shape of fungi. So the cell wall consists of carbohydrate layers, polysaccharides, glycoproteins, lipids, and glycans, but the unique component is a chitin. So, and the cell wall of the fungi is not that very well organized in a peptidoglycan. Some of the components can be analog to it, but it doesn't have this very determined structure with the cross-linked of the glycans with the polypeptides. Okay, so the structure is different. So it is not peptidoglycan. Then, apart from the cell membrane and cell wall over the cell membrane, the fungal cell may be covered from outside with the polysaccharide capsule, and that is a property common also in bacteria. So, and then the surface structure of the fungi, the membrane and the cell wall and the capsule may have a very important role in the pathogenesis of the fungal diseases. Why? What is the function of these organelles? First, they mediate the attachment to the host cells or host tissues. For example, the chitin layer or cell wall layer or the capsule may do this. Then The surface structures of the fungi may interfere with the host defense mechanisms of the human body. For example, they are very good in activating the uh, complement system that occurs in the human body and has a protective function against a lot of microbes. And then when this system is activated, then fungi cause very strong inflammatory reaction. And also the surface structures of the fungal cells are antigenic and immunogenic. What does it mean? It means the antibodies will be produced in our body against the surface antigens of fungi, that is immunogenicity. And these antibodies will react with the surface structures of the fungal cells. And also some yeast and molds may have some particular substances in a cell wall that may mediate some pathogenic reactions. For example, some yeast and molds may have melanin in the cell wall. So it has been associated with the virulence of these pathogens. Uh, Physiological characteristics of the fungi. According to the oxygen requirements, they are obligate aerobes or facultative anaerobes, right? It means that they may live mainly in the presence of the air, and only some of them may live in both conditions, aerobic and anaerobic. According to the way the way of nutrition, they are chemotrophs like bacteria. So, and how do they perform this nutrition? They secrete enzymes that degrade a variety of the organic substances into soluble nutrients, and then these nutrients will be passively or sometimes actively taken into the cell, just like in bacteria that also may perform active and passive transportation. Regarding the ecology of fungi, mostly they are saprophytes. What does it mean? I hope you remember. That use dead organic matter and don't need to live in the living body, alive body. But then some of the fungi are parasites. So those usually may cause human diseases, but sometimes, and we will mention it, saprophytes, when they get access to the human body, also may cause the human diseases. And the diseases caused by fungi are called, you can see here the term mycoses. So now we take a look at the morphology of fungi. And according to their appearance and according to their morphology, we may classify them into two groups. And you can see here these two groups, yeasts and molds. So, and we briefly discuss these both groups. First, we start with the yeasts. They are spherical or ellipsoid, right? You did see it under the microscope, and they are larger than bacteria, from 3 to 15 micrometers in diameter. And usually, most of them reproduce by a sexual process that termed blastoconidia formation or budding, but also may be reproduced by fission. Some species may produce, and you can see here, when they grow very quickly, may produce so-called pseudohyphae. When the oval cell grows very quickly, so the resulting cell will be not spherical or oval, but it will be elongated. So, and the elongated cells may stay attached. So then, when they stay attached, they form structures that are usually termed as pseudohypha. Why pseudohypha? Because it consists of buds that fail to detach, but every bud is a one single cell. Because yeasts are not multicellular organisms, they are unicellular organisms. So each cell here, each elongated or every elongated cell here is one single organism. So, and in the characteristic of the fungi, the microscopic morphology of the cells and macroscopic morphology of the colony. See, we see by eye the um, characteristics of the colonies. So, like you remember, they are creamy, circular, usually white, soft, opaque, cream colored, and the colonies are 1 to 3 millimeters in size. And that is very typical to fungi that are yeasts. So, and because they are larger than bacteria, yeast species may be identified on the basis of physiological tests and key morphological differences. For example, the types of pseudohypha, how they are the design of pseudohypha may be very important in identification, for example, of candida, in differential identification. Because, for example, the design of pseudohypha of Candida albicans will be absolutely different from the design of pseudohypha of Candida tropicalis. Okay, we go ahead. Now, a few words about the molds. So, and molds are multicellular filamentous organisms. And when they grow on the medium, they form multicellular filamentous colonies. That's what we see. That's what we see, for example, with penicillin and Aspergillus when it grows as saprophytic organism. And then when we take a closer look at this fragment of the hypha, and we don't call them pseudohypha, they are true hypha. So these are true hypha, branching cylindrical tubes. You can see here that these branching cylindrical tubes are not separated to one fragment for another one. So there is one multicellular organism. So the diameter of the hypha is usually from 2 to 10 micrometers. And sometimes they can be branching. And you can see these branching cylindrical tubes that have a common cell wall. See, the common outside envelope, the common cell wall. So, and I would like you to come back to the previous one. See, and pseudohypha, that's what we call pseudohypha, have cell walls that separates this single unicellular organism from this unicellular organism. Okay? So then, when we have the mass of hypha all together, they form a structure that is called mycelium. So the colony of the fungus is really a mycelium. Yes, and some hypha can be divided by septa, that is the cross walls, but they don't give complete separation. The cytoplasm and even nuclei may move within this septa and within these cells from one cell to another one. So multicellular organisms. If we take a look at the colony of the mold, we may see two types of mycelium. So the first type of mycelium is the substrate of vegetative mycelium or hyphae. What is it? These are the hyphae that penetrate the supporting medium, the where it is fixed on, and through these vegetative or substrate hyphae, uh, fungi absorb the nutrients, and also the vegetative or substrate hyphae or mycelium fix the colony on the surface of the substrate and not let anything to remove it from the substrate. That is very important. And then the second portion of the hyphae arise or project above the surface, above the substrate of the mycelium. And that's because they have a different function. We call this part as aerial hyphae. And the aerial hyphae usually is not for fixing in the substrate or absorption of the nutrients. It has a reproductive function. So, and that's because on the top of this aerial hyphae, there are some structures located that bear the cells that can be used, the spores usually, that can be used for reproduction. So, but it's not that simple. So we mentioned that we have yeasts and molds. But the thing is that we may have dimorphic fungi. What does it mean? It means that they may change the design of their body, the shape, the morphology of their body. So the dimorphic uh, species of fungi are capable of growth as yeasts or molds depending on the environmental conditions. For example, some appear as molds under 20 degrees Celsius, and you can see how they appear. And the mucor is the example of this dimorphic fungus. See how it looks under 20 degrees Celsius in its natural environment. But then we believe that when they get into the body, with 37 degrees Celsius, or in a laboratory if we grow under these conditions, they convert their shape to the yeast unicellular cells, right? And it is believed that, for example, in the human body, that's mainly when they may do this. It is easier for them to spread in a body via bloodstream or lymphatic, right? So, mostly dimorphic fungi are pathogenic. So, the dimorphism is considered as a very important pathogenicity factor. And here you can see some examples of dimorphic fungi that have this uh, mold shape under 25 degrees Celsius and convert into the yeast-like structures, and some of them, for example, coccidioides fungi, may form in the human tissues like a spherula, the structure called spherula that is full with or filled with or packed with the spores, where each spore cell is the individual organism. So later on we will take a look at the human tissues with this spherula. Okay. So now mostly they multiply by spores, and that's also the difference in between bacteria and fungi. Because bacteria, one bacterium, I would like to remind you, form one single spore. So the bacterial spores is just to survive unfavorable conditions, but not multiply. whether the spores of fungi, they are specialized structure that also will enhance the survival value of the organism. But they can be readily dispersed and they are more resistant to the adverse conditions, but then they also are used for reproduction because one single organism may produce billions of spores in its life, right? So then those spores, also when they are gotten into good conditions, will germinate and grow if conditions are favorable. So, in fact, these spores in fungi may be produced sexually or asexually, but that is important maybe for biology or the fungi. It's not very important for pathogenicity and pathogenic properties. So then, we mentioned that the reproductive structures are different, and the reproductive hyphae that arise from the substrate may be used as the properties for differential diagnosis of fungi because they are all different. So that's why we take a bit of attention, we pay a bit of attention on these reproductive structures. They can be broadly divided into two types. Conidia, this is the first one, and uh, conidia, where do we have them? Between, because they can be seen by two, they can be produced by two ways. So, first way, that is example of Aspergillus flowers. You can see this is a mold and these are the uh, aerial part of aerial hyphae. So, and at this aerial part of hyphae is has the very special structures at the top. And they produce phyalids, and this is a vase-like structure with the special conidiogenous cells. Why conidiogenous cells? Because these cells will multiply very quickly, and the result of it multiplication will be conidia or spores fixed on the surface of these conidiogenous cells. That's why the special specialized hyphae that have this structure, that bear this structure, is called conidiophore. So you can see conidiophore with phyalids and the conidia spores on top. And conidiophores and conidiospores are not covered from outside with any envelope or with any sac. So when conidia spores one by one, as soon as they mature, they will be released to the environment. And this is a picture or simplified scheme, and that is a photograph of the Aspergillus conidiophore and conidia. This is conidiophore with phyalids, and this is conidiospores, right? And that is example of the conidia asexual spores of Aspergillus and Penicillium. This is also conidia spores. But then, conidia can be unicellular or multicellular. If we have multicellular conidia, they are called macroconidia. That you can see that what is macroconidia. This is a large septated structure, and in each box in between septa we can have as separate spore or separate conidia that will be developed in one single organism. And the shapes of microconidia in different species are different. So that's, for example, microconidia of Candida, and this is macroconidia of Microsporum, and they are different. So the appearance of macroconidia can be used also in differential diagnosis. Second way how asexual spores are produced is when they are produced in Sporangius pores. You can see here still this is the vegetative mycelium, even with rhizoids that are special hyphae that extend into the substrate and absorb nutrients and fix the colony in the substrate. But then here you can see the hyphae that arise or project from the substrate and form sporangium. That is the sporangium uh, is an enclosed sac containing spores inside. And the hyphae that bear the sporangium is called sporangiophore. It is a specialized hyphae supporting the sporangium. And sometimes the hyphae may not be septated like in Rhizopus, it is shown here. So, and then you can see that spores are covered with, in sporangium, are covered with the sac-like structure, and the spores will not be released one by one. As soon as they mature all together, the sporangia for the sporangium, the sac-like structure will rupture and sporangiospores will come out of it and spread in the environment. So now let's take a look at the classification. We are not really very interested because there's a lot of species and the classification of fungi a little bit complicated. But we just mentioned the basic large taxonomical groups that are classes of fungi, and we will underline in which of them we have pathogenic fungi and give examples of pathogenic fungi. Oh, okay. So the first group is Zygomycetes, and they have both ways of reproduction, sexual and asexual. Sexual, it means with the help of meiosis, and asexual without meiosis, right? And so they result in zygospores development, and asexual way is formation of spores through sporangia. It means in sac-like structures. Hyphae are usually not septated, sometimes septated, and here we may see Rhizopus, and sometimes Mucor may cause disease. Then second group is Ascomycetes, where ascus is a sac. So they also have both types of spores production, sexual, and this happens in a sac in which karyogamy and meiosis occurs with production of ascospores. Asco because in a sac-like structure. And asexual spores are produced via conidia spores. It means not in a sac, but freely on the top of the conidia spores. And usually macroconidia are produced. And the hyphae usually septated, and the examples are, my dears, all of these are pathogenic. Blastomyces, histoplasma, trichophyton, microsporum, with the exception of Saccharomyces that are non-pathogenic. So next group is Basidiomycetes. And they again both sexual and asexual way of spores production. So, and the sexual take place again in a sac-like structure, basidium, and the result will be four progeny haploid basidiospores. An asexual way of spores production occurs in a form of budding yeasts. And usually hypha have complex septa. And the example of this, for example, in this group we may have fungi that we usually cause mushrooms, right? So that have these bodies. But then out of the pathogenic fungi, we may have here Cryptococcus neoformans. And that's how it appears in the cerebrospinal fluid in a form of yeasts. Then the last group is so-called fungi or fungi imperfecti. We call them imperfecti because the sexual way of spores production is not known and had not been observed yet. And the only asexual spores can be produced via conidia again, right? Not in sporangia spores, but in conidia spores. And the second name is Deuteromycetes on fungi imperfecti. The hypha may be in some of them that are molds septated, and in yeast, pseudohypha can be produced. And here you can see pathogenic examples that may cause very severe diseases. We take a look later on at them. Okay, now because we are studying not just biology of fungi but medical microbiology, it is important to understand how they cause disease in human being, and that is called pathogenesis. So we won't discuss it in very details. We just underline the groups of pathogenic factors or properties of these fungi via which they may via what they may cause damage to the human body. And roughly we can categorize all those pathogenic factors or fungi into three groups. First group, fungi may perform colonization of the human body, and those factors are called help in fungal colonization. So that's, for example, how we can see fungi in the aspirates from the human lymph nodes inflamed as a result of fungal growth there, right? So, but then they can grow in the human body due to some specific factors that allow them not to be killed, for example, by immunity in the body. This is one group of the factors. The second group is ability to produce exotoxins and release them into the substrate. The diseases caused usually by these fungi are called Mycotoxic Causes. So this is intoxication with fungal exotoxins. And finally, we may have the group of fungi that may cause hypersensitivity reactions in the body when inhaled or come in contact with the surface structures of the skin, with the surface of our mucous membranes or skin. So, and they may not grow in the human body in that moment, but they just cause hypersensitivity reactions. Okay, now one by one we take a look at these types. So the first example of mycotoxic causes. So this is the ergot producing parasitic fungus that is called Clariceps purpura. And the substance, toxic substance, is an ergot. You can see the Sporangios there and the uh, uh, spores that undergo maturation. So they may produce alkaloids and other toxic components, and then they can cause mycotoxic causes due to the ingestion of the fungi that produce this or the substrate where these fungi grew. For example, ergot alkaloids are produced in a grain if they are infected with Clariceps purpura. And the symptoms of these mycotoxic causes can be very severe, accompanied usually with very strong inflammatory response in the tissues. Then, with the necrosis, sometimes it may demonstrate neurological effect with hallucinations, and sometimes it may cause gangrene. So the second example of the toxins that are produced by fungi is, for example, the Aflatoxin produced by Aspergillus Flowers. And Aflatoxin categorized as a group of so-called bisphoranocumarine metabolites, and they are very toxic and highly cancerogenic. And initially they had been found as a causative agent of the disease in 1960s, in the early 1960s, as the cause of the turkey eggs disease in poultry. So, but then it was investigated that poults consumed contaminated uh, grain and develop lethargy, anorexia, muscle weakness, and necrosis of the liver, followed by the death. But also, these may occur in humans also. So then, second group of diseases or pathological effects attributed to the fungal infections are hypersensitivity reactions. So airborne fungal spores and any of the fungal parts may cause very strong antigenic stimulation of the body, production of the antibodies, and exaggerated immune response will be responsible for so-called hypersensitivity reactions. For example, if do you know, for example, those who prepare cheese workers? They used to inhale and have contact with fungi that are used to mature the cheese, right? So that typical um, disease that is connected with their occupation, so-called occupational disease, is hypersensitivity pneumonitis as a result of inhaling of the elements of the fungi. So, and usually the clinical manifestation of hypersensitivity towards fungi is rhinitis, bronchial asthma, alveolitis, and various forms of atopic reactions. And this is an example of the ringworm of the body caused by trichophyton. And we can see the very red border of the rashes that tells us about the type 1 hypersensitivity reaction towards the fungal elements. So, and then now we are talking about third group of pathogenic factors of fungi that allow them to grow inside the human body, to grow inside the tissues, for example, or to spread through the bloodstream, whatever. So, and of course here, like we mentioned the enzymes that help bacteria to cause diseases. Also, we may mention some enzymes that help fungi to grow in the body. But then here you should understand that bacteria are quite simple organisms, and it is easy for us to understand the particular role of each enzyme in the pathogenesis. For example, do you remember we used to say that hyaluronidase just um, break down the hyaluronic acid of the connective tissues, right? Or collagenase just break down the collagen fibers. But the fungi are much more complex organisms and they can produce much more enzymes. So sometimes it is difficult to trace the role of each particular enzyme produced by fungi in pathogenesis. But we may give some examples. For example, dermatophytes may produce uh, the enzyme that is called keratinase. What it does, it hydrolyzes the structural protein keratin. So the production of this enzyme help the fungal colony to be fixed on the surface of keratinized tissues, right, with the ability to lyse keratin, and also may help to digest it and to get some nutrition from it, right? Then the second example is specific morphogenesis. It means dimorphic fungi. Sometimes we think that, and that is, we believe, suitable for description of yeast pathogenesis. They may perform so-called filamentation. So we mentioned that the Candida may form pseudohyphae. And this filamentation that we see here, right? So they form filaments instead of oval unicellular organisms. This morphogenesis is important for the virulence, but it is not clear why, how really it makes it. So, but then generally dimorphism is very important because we think that for some molds that will be converted to the uh, spherically shaped candida like or yeast like structures in the body. For example, in that shape, that will be easier to spread through the bloodstream that we mentioned already. So, and elicit the stage of parasitism in them. Okay, so and also the production of the heavy capsule that you see here. See, this is fungus, Cryptococcus neoformans itself, and the yellow thick layer around is a capsule. And the capsule is sometimes thicker than the organism itself. And we think the capsule is important in colonization of the tissues because it acts as a for attachment of the microbe to the human tissues, but also they are important in withstanding phagocytosis, right? So this is the same. You've heard about some bacterial capsules in a lecture, infection and infectious process. So the Cryptococcus neoformans elaborates protective acidic mucopolysaccharide capsule that surrounds the cell and prevent elimination of it by macrophages. So now, as I said in the beginning, we supposed to categorize the diseases caused by fungi into several categories. But truly, they may be categorized in many ways. But we would prefer, for example, they can be categorized as exogenous and endogenous, right? So exogenous mycoses are those that are caused by fungi, natural habitats of water, soil, and debris, right? or taken from another person, while the endogenous mycoses can be produced by fungi that are part of normal flora of the organism. For example, candidiasis can be produced by candida that is a normal flora organ of the organism. So then so the main subdivision that we use here is the mycoses classified according to the place where they cause disease or where they cause infection. Okay? So take a look. So we divide them into superficial that infect the outmost layer of the human body, then cutaneous that go deeper, subcutaneous mycoses even deeper. Then we have a group of systemic mycoses. We call them primary systemic mycoses. What does it mean, systemic? It means they cause not local infection, like, for example, cutaneous or subcutaneous, but generalized and spread within the body through blood or lymphatic. And also, one category of the mycoses is opportunistic mycoses. These are mycoses that are usually accompanied with immunodeficiency or immunosuppression over the host organism. So those opportunistic mycoses, opportunistic fungi need some special conditions to be developed in a host body. Only then they will be able to cause disease in there. Understood? And finally, those that we discussed already are mycotoxic causes, right? The diseases that are caused just by ingestion of the toxins. Now let's take a look at them step by step. Superficial. Those mycoses are limited to outmost layer of the skin. And also they may occur on the surface of the hair. So, and you can see how they appear on the surface of the bulbs, right? And the causative agent is Tinea nigra. So, but then those fungi cause rather some social problems. Those mycoses are not very severe, and usually hypersensitivity reactions are not occurs in superficial mycoses. That is another example. So that is called Piteriosis Versicolor. There's a clinical presentation of it. If you take a closer look at the male's chest, you can see some discoloration of the skin. The agent that caused this infection grow only on the outmost layers of the skin or cuticle of the hair shaft. And they almost never induce severe immune reaction. And even if it is not visible, the changes that occur in the body. But if you take a scratch out of the infected area and perform staining with periodic acid shift stain, then we see the yeast-like and the hypha fragments over the fungus Malassezia furfur. See, we don't even see that it is there. Staining will help us to see it. Then, when mycoses go deeper, they cause when the fungi go deeper, they cause cutaneous or dermatomycosis or cutaneous mycosis. So they extend deeper into the epidermis usually than the superficial mycosis agents invade hairs and nails usually. And the example of them are Microscosporum species, Trichophyton species, and Epidermophyton species, and also Candida albicans and different other species of Candida may cause nail infection that are called onychomycosis and the hairs infection. And you can see how dermatophytosis may look like. There's a ringworm of the head caused by Microsporum, right? And cutaneous mycoses, because they go deeper, they cause hypersensitivity reactions. So because they have more strong invasive properties. So they cause strong inflammatory reaction from the host. So this is also example of cutaneous mycosis of the hair, and usually we may divide them into two types. Endotrix infection, where the spores are located inside the hair shaft, and this is typical for trichophyton. And ectotrix, where the spores will be located on the surface of the hair and freely spread. So, and this is called by Microsporum canis. Then, when fungi go deeper, they cause so-called subcutaneous mycosis. And usually the infection occurs through the damaged uh, so when the skin is damaged, so usually they won't occur on the surface of the uh, uh, skin not damaged. No, the skin supposed to be injured, or fungi should be inoculated deeper inside the subcutaneous tissues. And the infection may progress to muscles, subcutaneous tissues, and even fascia can be affected by these fungi. And you can see here the names of causative agents. And Sporotrix is maybe the very common one, and Madurella mycetomatis. This is a very common in India and is almost endemic for India, caused by mycetoma and some others. There's a quite severe diseases. You can see the disease caused, mycosis caused by Sporotrix, and it is common, very long term, usually chronic disease marked by skin ulceration and bumps formation under the skin. But then it may progress to bones, lung, joints, or muscles. So really can be severe. And that is endemic disease for India that is called Maduromycosis, also called Madura food, caused by Madurella mycetomatis. It is called mycetomatis because this is like a tumor, because it caused deformation of the tissues. But of course it is not tumor. But the body reaction towards this subcutaneous mycosis is very strong. So, and that is very severe disease. So, and then we come up to the systemic so-called primary or endemic mycosis. Usually they start with the inhalation of the spores and originate in lungs primarily, and from lungs they can spread to many organs via bloodstream. In histoplasma, blastomyces, coccidioides, and paracoccidioides may cause these diseases that are called those mycoses by the name or genus name of the causative agent. For example, Histoplasma capsulatum, and the disease is histoplasmosis. Blastomyces dermatitidis, and the disease is blastomycosis, and so on. So those blastomycoses, the systemic mycoses, are very severe. So usually, the respiratory tract is the first involved in the disease because usually we inhale it. And usually the multiplying agents possess unique morphological features. Uh, that is the example of Coccidioides immitus infection. So the spores are carried on the dust particles, and by the wind they can be spread within the environment, and they may be inhaled. So initial symptoms may resemble the common cold because that is not so strong inflammatory response. But then later on problems will grow, and they may develop within weeks to years, and that will be low fever, weight loss, bluish skin, breathing difficulties, and when they are spread through the bloodstream, the elements of fungi may be found in the blood, in the synovial fluid, in the spit and in bones and joints. And that's what I mentioned initially, that in these tissues of the host, we may find this ferrula, right? The structure filled with endospores. When they rupture in the body, the endospores come out and spread through the bloodstream. So, and this is another example of primary mycosis, that is blastomycosis, and it is caused by yeast-like fungus, Blastomyces dermatitidis. And except for the skin and subcutaneous tissues, it may invade and affect lungs, kidney, nerve system, and bones, and it may be lethal also. And the last one is the group of uh, opportunistic mycoses, and it is associated with immunocompromission, and they may be both exogenous and endogenous. For example, if the one inhaled the spores of Aspergillus or Penicillium that live as saprophytes, they will be exogenous fungal opportunistic infection. But also Candida species may cause endogenous fungal opportunistic mycosis. So, and this is the example of quite common one in immunocompromised individuals, especially those with HIV, that are HIV positive. So, and usually aspergillosis is an exogenous infection caused by different species of Aspergillus. And you can see in a chest film the cavities that are formed in the upper right lobe with an organizing fungus balls, so-called aspergillomas, right? So, and uh, they are capable of affecting any organs in immunocompromised individuals. But then, when it really happens in, for example, HIV positive individual, they can be uh, uh, lethal. That is usually fatal disorder. If we take a specimen, for example, sputum or blood, we may see the Aspergillus nidulans, for example, the structures of fungi spread by these tissues. So, and then there's a growing of Aspergillus in tissue. So these are molds, right? So, by the way, aspergillosis is not dimorphic fungus. It grows in the human body as a mold also, right? And we can see here septated hyphae in the tissue specimen taken from the body. And this is opportunistic candidiasis, except for quite common oral thrush that occur usually as a result of overexposure to antibiotics, long-term antibiotic treatment, because those who undergo long-term antibiotic treatment against some bacterial infections, those drugs may not affect the fungal growth. So, when bacteria growth is inhibited, the fungi may grow very well and overgrow and spread very fast. But then they may cause also other diseases like what's this? Я читаю лекцию. So this is usually produced in immunocompromised hosts, and also quite often they may spread through the bloodstream and cause life-threatening disease, fatal disease. Now let's take a look at briefly how to perform laboratory diagnosis of the fungal infection. Usually, if we are talking about primary pathogens, we mention them, right? Those that may cause systemic mycosis. The diagnosis can be made with the help of microscopic examination, and we need to visualize the morphological structure of the organisms. That's very helpful because they are large, it's easy to see the differences. But also we may perform the culture, and usually several agar is used for their growth. Then the diagnosis of opportunistic agents like aspergillosis, candida, mucor usually are more complicated, and we have to perform culture isolation, and we have to perform microscopic examination to find the fungal elements in material. So usually we also may use PCR or immunological tests for species identification of these fungi. So, and the isolation is not so different from what we have for bacteria. Again, so we can see, we just repeat that we may see agar plate for the separation of the colonies, right? So the broth is used to increase the growth of bacteria. Then from the broth, usually if we can't start with the broth, we perform separation on agar plate, from agar plate usually to agar slant where we keep a pure culture, right? And the stabbing inoculation in some agar columns or liquid media that we use for identification. So this is strict culture. I just want to remind you, this is strict culture of fungus because in candida, because we see creamy round colonies, and we keep them in the straw culture as a pure culture. And then we use the same approaches in the identification both of bacteria and fungi. So microscopy, that is more important for fungi than for bacteria. Then the cultural characteristics means how fungi grow on the slant or on the plate agar or on some other media. Okay, then the biochemical tests are used because fungi also perform sugars fermentation, and that's the very common way how we identify fungi. Then we may perform biotyping, serotyping, and phage typing. That's same with bacteria. And if you are talking about microscopic identification of fungi, you can see these are the different structures of the reproductive portions of aerial mycelium. And you can see the conidia spores and sporangius spores and how they appear. And we can see different types of spores and conidia spores. So this is really very important in identification. There's a colony morphology and all this. But then additional test is the uh, antimycotic resistance. And also we can use serotyping, genotyping, and that. Now I would like to stop at the antifungal agent. So we said that there is difficult to find a drug that will act selectively against eukaryotic cell of fungus, but doesn't cause any severe damage to the human cell. And that is a problem to find this type. So then, generally saying, if we take a look at this, so as I said, this is a simplified scheme of the fungal cell, right? So if this is the cell membrane, so we have a very good group of drugs that are called polyenes. By the way, they are also antibiotics produced by other living organisms. And two are the best, nystatin and amphotericin B here. Then we also have a lot of so-called azoles that also has a target as cell membrane. Then we have a group of drugs that are called echinocandins that have cell wall as a target for anti-fungal therapy. And then we have nucleic acid synthesis as a target for antifungal therapy. Now we take a look at these most common types of drugs used as antifungal agents. So polyenes affect the ergosterol by producing pores in the cell membrane directly. So these drugs bind with the ergosterol and change its uh, structure and make the pores in the cell membrane. So by now, amphotericin B is recognized as the best one, and it's been used in treatment of very severe systemic mycosis. But then it is nephrotoxic. So then nystatin is used to treat the local candidiasis. Administration also can be oral or local. So, but no systemic absorption occurs very good. So then, azole derivatives, many of them, fluconazole, ketoconazole, intraconazole, and the others. So they also affect ergosterol, but not itself, ergosterol itself, but synthesis of ergosterol, because those drugs inhibit the cytochrome P40, 450 dependent enzymes. And of course, they are a bit toxic for human body. They usually broad spectrum anti-fungal drugs, and they may be used systemically and locally. Some of them are too toxic to be used systemically, like clotrimazole, some imidazoles, and others. And some of them can be used systemically. So then we also may use nucleoside analogs, and example is 5' fluorocytosine. And it interferes with the DNA or RNA synthesis, inhibit this. And they are usually narrow spectrum, but they are effective against Cryptococcus neoformans in systemic cryptococcosis and in candidiasis. They are not so toxic, but the resistance is produced very quickly. So then we have a group of so-called grisans, griseofulvin, that interfere with the production of and function of microtubules. They are of narrow spectrum and used against dermatophytes only. They are not highly toxic, but the therapy may take from weeks to months. Okay, my dears, because we work without the break, with this we may finish our lecture. And at the next first week, we will continue our special microbiology lectures. Thank you for coming. So you may be free as birds right now. Goodbye.